Hey everyone, welcome to our second video about protein synthesis. This is going to be an overview of how proteins are made, and we will get into more specific parts in our third and final video. So check this out. Usually I actually do this lesson by drawing on the board, and I don't typically have it on a slideshow, but I am working with it this year, so bear with me. So this is protein synthesis, or how to make a protein. This is a cell that I've drawn here. So the cell also has a nucleus, and the nucleus has chromosomes and DNA in it. And what's going to happen in protein synthesis is that the DNA from a chromosome is going to open up with helicase. You can see that there happening in the nucleus. And mRNA is actually going to be copied from that DNA. The mRNA is then going to slither out of the nucleus and into the cytoplasm, where it's going to meet up with a ribosome. That ribosome will then kind of hold on to the RNA in chunks of 3 that are actually called codons. And then those chunks of 3 are actually going to code for a specific tRNA. You can see in blue there, tRNA molecules. So the tRNA molecules are actually pretty cool because they carry amino acids on them, and those tRNA molecules will carry the appropriate amino acid to the ribosome to form the protein based on what the code is on the mRNA. And remember, the mRNA came from your DNA. So basically, DNA gives the instructions to mRNA, which mRNA carries the instructions to the ribosome. The ribosome then is a spot where the tRNA grabs onto the mRNA instructions and delivers an amino acid. And for every 3 nucleotides on mRNA, it's called a codon, and it will code for 1 amino acid. Those amino acids start to build up, and you can see that here, they're going to make a protein. So these processes are actually 2 steps that are taking place. The first step is called transcription, and that's when DNA actually opens up and serves as a template for mRNA to be made. And that occurs in the nucleus because the DNA actually has to stay in the nucleus no matter what. It is too unsafe in our cells for DNA to come out of the nucleus. It is quarantined in there, if you will. So DNA goes through transcription, where a copy of mRNA is made from the DNA. And the mRNA then, because it's only a single helix and it's much smaller, it's able to slither out of the nucleus into the cytoplasm, where it's going to meet up with a ribosome for the translation process. And in translation, we have what's called a codon, and we use a codon chart to be able to figure this out. But we have every 3 pieces of mRNA are part of this big code for making the protein, and every 3 pieces is called a codon. And every codon actually codes for or tells us which amino acid will be brought to the ribosome by a piece of tRNA. And those amino acids will then bond together. And when you have a bunch of amino acids, they form a large folded protein. So that's kind of a big overview with a drawing that I would normally draw on the board for you, but hey, it's online this time. So here is just another way to think of it. Protein synthesis is a 2-step process. The first step is transcription. Transcription occurs when DNA has a gene from a gene is copied to produce an RNA transcript that is called messenger RNA or mRNA. So transcription gets you from DNA to mRNA, and it happens in the nucleus. Step 2 is called translation. That's when mRNA is a template for tRNA, which brings amino acids together at the ribosomes. This happens in the ribosomes. Here's another way to look at it, kind of a big picture. So we start with DNA that's in the nucleus. DNA cannot leave the nucleus, so it goes through transcription to make strands of mRNA. Those pieces of mRNA are able to then slip out of the nucleus into the cytoplasm, where they will meet up with a ribosome. When they get together with a ribosome, they do translation, and that calls for certain amino acids because every 3 pieces of mRNA are what's called a codon, and they code for a certain tRNA to bring a very specific amino acid. Those amino acids fold up in the cytoplasm to make a folded protein that can then be used by the cell. Thank you so much for listening. Next up, we'll talk about the more specific parts of transcription and translation. But thank you for listening about the overview of protein synthesis.